Like, I looked up yeah, yeah. It's like a, there's like a fun and like
Guys, we're gonna get started, guys. We're still waiting for Nick to come upstairs, but he'll be here in a few minutes. In case you guys are wondering where Nick is at. But in the meantime, it's my great pleasure to have Colby come to visit us. He's apparently flew all the way here just to talk to you guys.
Wow.
Pretty cool. So he's a computational biologist at Tupel, specializing on building cloud-based solutions for life sciences. He's also apparently an author of a book, Genomics in Azure Cloud, published in O'Reilly 2022. Not sure if I just get this part, but also the most valuable professional awardee.
I sent him my my bio that's just a standard bio, and I told him to cherry pick from it. See what he cherry picks.
I know that's pretty cool. Okay. Also visiting scholar at Charlotte's UNC. All right, I think I'll stop with it. All right, well, Colby, take it away.
Sure. Hi everyone. My name's Colby, and I will be talking about DevOps, but not like the boring kind of DevOps of writing code and pushing it to GitHub. I really want to start talking about how we use standard practices that are popular outside of the sciences, but for scaling up and deploying the models and tools that you're used to using today. So before we get started, just a couple disclaimers. So everything in the talk is public. Feel free to take pictures. It'll be online, obviously. All the opinions are my own, but some of the work is collaborative with MIT Lincoln Lab, as well as some professors and other researchers at UNC Charlotte. And some of the work is funded by 2 different grants, one an Air Force grant that you see, and one is from NC Biotech. So thank you for the funding while we still have it. Maybe I should behave, this is going online. A little about me. So I am the owner of a company called Tupel. We're a Microsoft partner consulting firm. So I do a lot of cloud architecture for companies like Seattle Genetics, which is acquired by Pfizer, companies that do pharmaceuticals or biotech work. And so I focused on building cloud architectures that scale really well for the sciences. And we have a subsidiary that we spun up that's called Silico Biosciences. It's more of an AI drug discovery sort of startup venture that is just in its infancy. But in addition, I'm also at UNC Charlotte in the CIPHER Center, which is our Center for Computational Intelligence to predict health and environmental risks. They paid a lot of money to some marketing person to come up with that acronym. But it's basically our infectious disease research center. And I'll talk more about that in a little bit. I teach cloud computing. No shocker there. And a lot of my research work is in infectious diseases, but also protein modeling and a lot of just scalable cloud and high performance computing infrastructure. I am the author of Genomics in the Azure Cloud, an O'Reilly book. And before you ask, no, you do not get to pick the animal that they put on the front of the book. All the animals are endangered species of some sort. Most people get lovely birds, and I got the Tamworth pig, which is a big brown pig. So while pieces of this talk will be Azure, I'll talk a lot about Microsoft Azure. Don't get up and run away if you don't do cloud today, or if you use AWS or GCP, because a lot of this work will be, a lot of this stuff will be applicable if you use a different cloud. I just happen to do a lot of Microsoft work. I'm a Microsoft MVP awardee for my work in bioinformatics in the cloud and AI, and then I'm a Microsoft certified trainer. And that is the last of the sales pitch that you'll hear from me. The rest of it will be just cool stuff. So some of my previous work is in a lot of structural biology surrounding things like SARS-CoV-2, and we looked at predicting the structure of various variants. And the hope was that we would provide a bit of insight before, along the lines of how well our current antibodies working, our current defenses working, and can we give the public health scene a bit of some insights way before the lab processes could be completed. So we started with Omicron and then did some subsequent work with XBB 1.5 and JN1. And then recently did some work with MIT Lincoln Lab on looking at human cytokine interactions with various SARS-CoV-2 mutants. Previously, I did a lot of work in malaria, and then most recently in H5N1 influenza. Most recently, a couple papers that we published includes a paper published in the Lancet eBiomedicine for a large-scale antibody binding predictions, and then a paper just got accepted a week and a half ago or so that's on antibody diffusion, so designing new antibodies in general, but the candidates that we released in the paper are for the AJ1 protein specifically.
Colby, just to interrupt quickly, I don't think it doesn't sound like your mic is on. I just want to make sure that people can hear, or we're recording.
Can you hear me now? Can you hear me now?
Yeah, I can hear it on my.
Oh, it's all good? Okay.
Okay.
Yeah. Sorry for interrupting.
The light is on, whether anyone's home, I don't know. All right.
Oh, you mean like, can everybody actually?
I couldn't hear the mic. I mean, your voice is fine in here for me.
Can you guys hear me?
But I was just worried about Zoom.
Oh, no, Zoom's good.
Okay.
Okay, cool. So in addition to structural work, I also like to build a lot of tools. So things that people can reuse and without having to reinvent the wheel each time. So recently I published Pymol Fold, which is a plugin for Pymol that allows you to fold sequences directly in the GUI. So rather than going somewhere like CollabFold and then folding something and then downloading the PDB file and then opening it in Pymol, you can just do it right from the GUI. This works with Boltz, Chai1, ESM through the API, and then I think I'm missing a few. And but it also allows you to grab some sequences from databases as well. The antibody diffusion paper that I mentioned that just got accepted, that also includes a pipeline that's a bit adaptable. So you can provide an antigen structure and some reference antibody structures, and it will diffuse new candidates through an easy to use StateMake pipeline, and then give you an output that looks like what you see on the screen here. And then I also do other things that aren't related to structural biology, including things like an MCP server or model context protocol server, or that allows your AI models to chat with databases online. So for example, in this situation, I can ask the protein data bank, tell me about pembrolizumab, and it gives the description there. So this is more of a fun side project. I also have done a lot of work in the phylogenetics world previously and maintain a package called String Hub. It allows you to take phylogenetic trees, overlay them with metadata, and then produce these transmission networks that can help paint the picture of what are the patterns of transmission that we see. And this is especially effective in infectious diseases. So like what you see on the screen here is we see this for H5N1, we see this big efflux of avian to mammalian transmission, but not very much going backwards. But we do see some mammalian to bird transmissions. This is kind of
What does MCP mean?
Model context protocol.
For LLMs.
For LLMs, yeah. So it allows you to, so like if you're running like Claude locally on your machine, you can have Claude ask questions through MCP. It will select tools that are in the MCP toolkit that you build. So for example, one can talk to the protein data bank, and then it will execute code, get the responses back, and then the LLM is able to parse through those results. It's a pretty interesting thing, but it's becoming pretty popular among a lot of cloud vendors and a lot of just third-party vendors that want to make it to where you can use your LLM on their tool. So when I was asked to speak at this event, I looked at some of the other talks that were in the series and was thinking, well, I don't make a whole lot of models myself, though that's starting to change. But what I wanted to give you guys is something that is maybe will help you use those models that you learned about in the other talks, but use them at a larger scale, use them in a way that's reproducible, and then in a way that you can share them with other people. So today we'll be talking about modeling, packaging, and deploying. Starting with modeling, how do we train and tune protein language models, and where do we do that? In the packaging, how do we make your model or tool usable by others? and without making it where you have to follow a 75-step process to get it installed. And then once we've got everything packaged up, how do we deploy it? And what are the options to do that? And whether it's deploying so that someone else can use the tool, or it's deploying so that you can scale it because you want to do 10,000 experiments, I'll show you how that's done as well. So let's start with modeling. So let's say you have the idea, you wake up at 2 a.m. and you're like, oh my gosh, I have this new amazing idea for a new model. Should I, A, build it from scratch, or B, tune/adapt an existing model? I think most of you are probably selecting a letter. The actual answer is neither. Step one is you got to get your data first, right? And this is the problem with any sort of AI project, but especially in the structural biology world where there's not enough data. So if we think of models like GPT or any of the Claude models, these giant large language models, they are trained on billions to trillions of observations, tons of text information, and that allows them to generate these models that are really good at generating text. However, we don't have data sets like that in biomolecular space. So this is sort of a call for, as you're writing your grants and working with your professors, start making some good data sets, because it's what we need in order to do better AI in the biomolecular space. So just as an example, Protein Databank has about 238,000 structures as of the other day. So 238,000 of protein structures that are in PDB that are experimental, so a lot less than the billions that we see in the LLM world, right? And then in addition to the Protein Databank set, we also have the AlphaFold and Model Archive databases that include non-experimental structures, so predicted structures by various methods, and that's around 200 million. So still, even if we were to make predictions off of predicted structures, it's still not in the billions that we're used to in the other fields. In the small molecule space, we're a little bit better. Still not where we need to be, but we have lots of compounds in the Kimball and Pumpkin databases. But if we wanted to do things around affinity or molecular dynamics, then the data sets get even smaller. So for example, QM9 is one of the data sets that includes a lot of affinity information and also just biochemical information about the small compounds, but we only have about 7,000 records in that data set. So not enough for us to do that. Yeah,
the problem variety or quality of the data sets. As far as I know, PDB has some very good quality stuff, and what I know, PDB Bind just doesn't have very good content.
Yeah, so the question is the problem quality or quantity, right? And I think a lot of the problem is, as with any public data set, you get a lot of variety and inconsistency between records, right? How people put information in there. And oftentimes with a lot of these entries, we want the other metadata about it as well. So, for things like PDB, do we want information around like what is the protein called, and is that annotated with a nice protein ID or whatever? Like there's other metadata that would be useful, and a lot of times that doesn't exist without a lot of curation. And at the intersection of this, if we wanted to build a new model that works on complexes, so predicts multiples or protein-protein or protein-ligand complexes, Binding DB has about 3 million complexes, and then PDB Bind Plus, which just came out from a group in China, has 34,000 molecular complexes, but still not a whole lot. So where do we go? What do we do from here? And this is a thing of, well, if you wanted to train a model from scratch, you need a lot more data than if you were to just tune a model. So I'll give an example. So a recent project that we started at Silico Biosciences, the Tuple subsidiary, is we're building a suite of models called Polike-1 that are fine-tuned large language models that are tuned using antibody antigen data sets. So we first collect the antibody sequence information from SABDAB, which is the structural antibody database out of Oxford, where we pick some open source LLMs, fine-tune them, and then we're working on evaluating the results. Now, some of the astute of you in the audience are probably like, that's not an antibody, you idiot, what are you doing? So, but this was the first structure or the first sequence that came out that we folded, and this is what, this is what we generated first, which was really cool. It's not an antibody, but we did prove that we can at least generate a sequence of amino acids, or at least characters that look like amino acids that can fold into something. So
Colby, what are the examples of the open source LLMs that you were exploring?
yeah, I'll show you that on the next slide after this.
Okay.
So the data curation part is really the crux of this whole situation. So SABDAB is a subset of PDB that is annotated antibody and antigen interactions. And then from that, we then go and grab that information, and we've built out this sort of just flat data set of this.
Can you define what an interaction is? Is that like 1 millimolar, or, you know, 1 nanomolar? What does an interaction mean?
Yeah, so I'm not sure how SABDAB defines it exactly. It's just, does the antibody and antigen exist in the same space in the PDB file? We're getting ready to annotate this information with things like polar contacts and other like epitope information, and we'll set an actual angstrom length on that, like 3 angstroms or something.
But so I just think of it as a binary thing, bound, not bound.
All of the SABDAB entries are bound by whatever that, well, I don't know what their definition of bound is, but they're all complexes in the SABDAB dataset. So in this, we start with around 9,000 records in SABDAB. So now we're even fewer records than we had before. We go through each one, download the full PDB files for each one, and then filter down to the chains that are of interest, that heavy and light chains and the antigen chains, and then extract the sequences from that. When you filter out records that have one of these chains missing, or filter out duplicates, or things that are almost duplicates, then we get down to around 5,000 records. So now we're even farther down than where we really want to be. So then we stop here, and we're thinking, all right, let's just go ahead and throw this into the tuning process and see what comes out the other side. So one of the easiest ways to tune some of these open source models, so these open source models include things like Llama from Meta, the DeepSeek models, Quinn has a series of smaller language models, and then the one that we started with was Microsoft's V4 model. It's about 14 billion parameters. And we can pull these from Hugging Face. Who's familiar with Hugging Face? Most of you, okay. So for those of you that are not familiar, Hugging Face is an online repository where organizations can upload and host their models. It's really nice because they have a very easy to use API that will allow you to download the models and then do stuff with it, whether that's push new data through it and inference, or if that means if you wanted to fine tune them as well. So in this situation, they have a nice package called Transformers that allows us to pull the model. So you see here, I'm pulling the model and the tokenizer. And all I said was just pull me the Microsoft V4 model. That loads very easily. And then we have some tuning information that we wanted to do. This is pretty boilerplate code. So if you follow the tutorial, I didn't come up with a whole lot of this myself. And then also, without having to retune all 14 billion parameters, we can also use what's called a LoRa config, which is this a method that only it allows your tuning process to only tune a small subset of the parameters rather than all 14 billion or however many is in the main model. So in this situation, we pass all this configuration information in, and then we're able to train locally. So this is using PyTorch under the hood, but pretty easy to follow through. And in nowhere in this did I actually define anything about like neural network layers, multi-layer perceptrons, attention heads, none of that, right? All I'm doing is using the existing architecture and feeding it the 5,000 something pitiful records that I generated on that last slide. So then once we train, we only train for a few epochs because we're training locally for now. We start to see this plateau of the loss function. So basically, we're starting to see a plateau as for its performance, as to what the standard definition of loss is. But loss in terms of by the way, lower loss is better. In terms of loss as it stands with like a large language model, maybe that doesn't really apply when we're talking about biomolecular models. So what does good look like, right? So if we start to plateau around the 2 to 2.5 range for loss, if I were to keep training this for 30 epochs or 300 epochs, and it continues to plateau, does this get better? And the hope is that yes, it does get better, but what we're doing is we're starting to add in additional evaluation metrics aside from loss that may be able to tell us. So these are things like the antibody sequence that it gave us, should be a numberable? Does it follow the standard format for an antibody, right? how foldable is it? Like, do we get good confidence out of what was folded? Things like predicted binding affinity, or if we gave it an antigen and said, give me a new antibody, did it even bind there? Some of these metrics that we're trying to add in. And then also things for desirability, right? Is the antibody that it generated, how human is it? Is this something that could be turned into a drug or not? So 3 slides ago, I showed you that really cool blob structure that definitely wasn't an antibody. It was cool looking. And then after we did this very minor training, and we gave it this white structure here is the HA1 protein from bird flu. And we said, generate 4 more. And every single one of them folds into an antibody structure, is able to be numbered through the Tortilla system, and binds at least at face value to the antigen structure. So, yep.
So I guess it would be nice to know a little bit more about what is being trained. So from what I understand, like a protein language model, you feed it protein sequences, you mask some of the residues, you'd have it predict the masked residues. Here it looks like you're feeding it an antigen protein sequence. And then it seems like the task of, at least during inference, the task of the model is to produce a full antibody sequence or at least CDRs. During training, what is the task of the? Yeah, so the tuning process. So normally in a training process, you would give it a sequence with some things chopped out, right? And then it fills in those. In this situation, because we're tuning it, because the model already knows how to do like language stuff, you know, write a poem or a CV or something, or a cover letter. I mean, in this situation, we're giving it the antigen sequence, and it's producing these two, the heavy and light chain sequences, specifically the Fab portion, the Fab or FV portion. The full sequence. The full sequence. Not just the CDR. Correct. Yeah, the full sequence.
You didn't sort of prompt for that.
Say that again.
You didn't sort of prompt for that though. You give the LLM prompt saying that this is here, and the fine tuning basically rewards it
rewards it for getting something that looks like a heavy. Yeah. So this is the kind of the point of where I'm coming from. Didn't have to do a whole lot, right? I didn't really guide it to do very much, other than I gave it one column and I said, give me the other 2 columns.
What was the reason for starting with like an LLM trained on everything?
Yeah, because we only have like 5,000 records, right? So if I wanted to start from scratch, and we may still try this, right? But if we wanted to start from scratch, is the 5,000 records enough to even get the language model producing anything, much less? Because there's a whole lot of what happens inside of the attention heads of these large language model architectures that you just need to give it a lot of stuff to even get the parameters to a decent point of just producing anything, regardless of what it's supposed to be producing. And this is actually the same thing between, like, if you were to take a large language model trained on English and give it Chinese, it's the same sort of thing where you have to get to a certain point so that the parameters even just come up with a result that's not random. I don't know who was next.
But then, like, if you just like use not LLMs as the starting model, but like models that are like specific, like big models, not like LLMs, but big models that are trained for biomolecular stuff, and then tune it on the antibody.
Yeah, so the idea, so in addition to the V4 model that we're using, we're also going to look at some others. So there's a Nature LM that Microsoft just published like 2-3 weeks ago that has the notion of both language as well as amino acid and nucleic acid sequences. So we're going to try that as well. The limit, as in what we can do with this transformers library that makes it easy, is that you have to have something that fits within this. So for example, there's a language model called Lobster, if you've ever heard of that one, that's for amino acid sequences, but it does not fall within this paradigm. So we'd have to like figure out how to tune it manually, which we can totally do. But the point of this is to show how easy it is to do it with this transformers. Yep.
My question is related to data. Like for the protein, there are IDR regions, right? So like, would you suggest like, or like, will you tell us approach like how we can train an LLM which will identify the IDR regions for that given protein sequence, and it will also give us the molecule which reacts with that targeted region, like with that IDR region, in order to make that protein more stable and to stop the binding or forming cluster forming in a human brain or body?
Yeah, so this was the first pass, just see, can we generate something that looks like an antibody? Next, what I have, I have an intern that's working on this right now is from these individual PDB records, can we highlight the epitope residues, the active residues on the antigen sequence? So that in the future, you could say, here's the antigen, and I'm going to highlight the residues of interest, and the antibody that is produced is specific to that. Yeah, this is just the first pass. This is literally hot off the presses 2 nights ago. And then what we're working on now is just beefing up the information that's in these sequences. The other thing too is we also need to post-process some of these Fab sequences because they're not always full Fabs and they're missing some residues. And so we need to handle that a little bit. So data cleanliness is also important, and we're early on. Any other questions? Yeah.
Where did the structures come from that you were showing?
It's folded. It's AlphaFold 3. No one judge me. It's AlphaFold 3 folded versions of the alphabets. Okay.
Yeah, I think maybe a question that's sort of building off a couple of things that other people asked, but why not start with a protein language model?
Correct.
With the 20 amino acids,
Yeah.
tokens, and
So
generate.
So the two things. One is what I mentioned earlier about the ease of fine-tuning some of the protein language models that don't fit within this transformers paradigm, which we can totally do. This was a pet project that actually started having some success. This is to be perfectly honest. The second is the tokenizer is actually modified. I don't show it in the code here, but when we load in the tokenizer from the Microsoft V4 model or any of the other open source models, there's subsequent lines of code here that I don't show that modifies it to add in the other amino acid records, or amino acid tokens, excuse me. So it has the full 20 amino acids and then any of the characters that we add. So for example, we also wanted to use the pipe delimiter between chains. And also the chains, if the antigen has multiple chains, like you see in that first row, so we added the pipe delimiter as well. So the tokenizer is updated to be specific to amino acid letters. All right. So now, so far, we've run this model, this tuning process on a local machine. And I think if you ever want to get started on doing this yourself, you may think, well, how and where do I run stuff, right? Locally, it's a really good place to get your code running because it's a lot easier to debug your code in a Jupyter notebook or in VS Code than it is on an HPC system where you have to set it 1,000 times and then and your code doesn't run. So it's really useful. I think a pro tip is get your code running locally and on a small baby data set, on a small like 3 or 4 training iterations, and just make sure that it runs, and then make it work in the HPC environment. And then in the later slides, I'll talk a bit more about how to put things in the cloud and on HPC, but I think the question is, the cloud is really useful for if you need external collaboration. It's a lot easier for me to share something with another institution and share access to a cluster that way than it is for me to get them credentials to my university cluster and that kind of stuff. Because if you have a cloud account, you control the cloud account. If you have an HPC account, IT controls that, and we know how those guys get. So, no offense. But I do think that HPC is really useful from a predictability perspective from costs, but it is a shared resource. So once you have your model done, like let's say I've got this model up and running and I really like it, how do we package things up so that the models and tools are useful by others or usable by others? So on that giant slide of other previous papers I've published, a common denominator in this is this sort of workflow where we collect some proteins or target proteins or antibodies or whatever. We might grab the sequences out of them, or we might diffuse new sequences. And then we have this protein folding process, protein-protein docking process, and maybe we do some molecular dynamics on it as well. And then we get the outputs from that, whether that's metrics or the complex structures or trajectories or whatever. The thing is, this part is the complicated part, and that's the part that we want to perform concurrently on multiple complexes or multiple experiments. So as an example, the paper with MIT Lincoln Lab where we looked at human cytokines against the nucleocapsid protein, we had 64 human cytokines paired against 17 different mutants of the nucleocapsid protein. So that gives us 1,088 complexes. So that's 1,088 experiments. And then from each of those, we wanted to test the results of Haddock, or this is a biomolecular docking system that uses some physics-based modeling, so it's not purely AI-based. And then also AlphaFold 2 Multimer. And from that, so we're running lots of iterations there, and we're generating a ton of PDBs. And then after we generate the outputs, now we need to summarize that into something that makes sense. So picking the best structure or whatever. So we sort of have this experimental overload. So this part in the center here needs to be done 1,088 times. Part of this process is a bit blended. So some of the experimental preparation, like grabbing some data, getting the input files up and running, happen locally. And then there's the HPC part where we want to put things up on the HPC environment, submit a bunch of jobs, they complete, and then we grab the results and pull them back down. And then we filter out stuff that we don't need and throw away some results that we don't need, and then go and get the best results, whatever that means. And then maybe do some secondary metric calculations, et cetera. So the question then is like, well, how do you know what parts to do in the cloud, and how do you make this HPC part work really well? So as some other examples, the H5N1 antibody paper, that one had like 1,800 complexes. This is a student's project that's in progress right now. It's 10,000 ligands against one EB2 molecule, so 10,000 different experiments. And then I participated in the Adaptive Bio EGFR Design Challenge. And just for the first iteration of this, it was 200 proteins by one antigen, EGFR. And in that situation, everyone's proteins, you don't know where they were supposed to bind, right? So in situations like this, where we know the active residues in the CDR loops of the antibody, and then we know where the binding site is supposed to be, we can sort of control the experiment a bit better. And for like protein ligand interactions, we know where the binding pocket is, so we can sort of control that. But in situations like what you see, the hairy ball of wax that you see on the far right there, the experiments take a long time because it's having to check lots of residues. So one consideration to have whenever you're working on these experiments is trying to reduce the search space while not biasing your results. So as an example here, we could have started and said, all right, I want you to check where this other protein binds to any of the surface residues on the target molecule. That's the least amount of bias. I'm not trying to guide it in any way, but it's the slowest process. And then we can kind of start whittling down the site. And then I could go all the way to just to active residues. It's the quickest to run, but I'm forcing the docking systems to go right there. And that doesn't necessarily mean that that's going to be the lowest energy binding spot, or that may not even be accurate at all. And the system will just force something there and give you erroneous outputs. So you kind of have to be careful. So how do we scale this? Well, spoiler alert, containers. So we start with a container image that includes all of our models, the dependencies, the scripts to run things. And this makes that code very modular, and we can throw that up in a distributed environment, like an HPC environment or Kubernetes, which I'll talk about in a minute. And then once we get the results, then we can collect those results and filter down to the things that we care about. So containerization is key. And how many of you have ever made a Docker container? Okay, good. So I don't need to tell you that Docker is important. And I think that the biggest thing for those of you who haven't run Docker containers before is that they allow for portability and reproducibility of your code. And it makes it to where if your LinkedIn feed or your Twitter feed or whatever's like mine, where every week there's 5 new AI models that come out and you want to try them, and then you go to their GitHub and then you see the install.md file that's 17,000 pages long, and you have to have a new version of CUDA for some reason, and you have to uninstall your PyTorch version again, right? You don't want to do that. And so one of the big things, even if you don't ever need to scale what you're working on, it allows you to avoid messing up your local environment. So especially for some of these models where the authors act like your environment has to be exactly like theirs and has to run the same day and it can't be raining outside, right? Like we want to avoid that. And so it makes it a lot better if you provide a Docker container for your model that you publish, or if you turn someone else's model into a Docker container. Yep.
So just to let you know, I don't have a strong comp sci background, but I've noticed that Conda is not on here. I use Conda a lot.
Yeah.
Does that not count as a completely isolated container?
Yeah, so condas are environments. So it's like it's literally a separate path on your local computer that says, I'm going to use this conda environment called bolts2, and in that conda environment literally has the Python executable and any of the package versions that are in that environment. But it doesn't have other things that are related to running things. So things like drivers, like if you need a specific GPU driver or things like that, it's not a container because it doesn't run on its own. It's literally like just another version of Python. It's on the machine.
Okay, I see. Thank you.
Yeah. So the big thing about containers is that they work both on your laptop and your friend's laptop, but they also work in the cloud on Kubernetes and other containerization services, as well as on-premise in HPC environments. One big thing is that with Docker, it requires root level access. So a lot of cluster admins don't love that. So a lot of times in HPC environments, you'll see Apptainer or Singularity images, but it's very similar. And then the thing in the middle is called Podman. It's a more limited sort of version of Docker that does not require root access, but it's becoming popular for environments where you just need to run something that doesn't need all the bells and whistles. So if you haven't run a Docker image before, or have never created one, Docker images are built like layers of an onion. So you start from a base image. This is usually the operating system. So in this situation, I have an Ubuntu base image, and then I'm going to label it with some information, maybe whoever the maintainer is, or give it a description. I can install dependencies. So this is where you would install more like the system dependencies, things like sudo, things like a text editor, things like git or wget or curl or whatever. Define some environment variables, and then have some commands that actually do other things. So making directories, installing stuff, etc. This section is where you would put the conda environment. So you can use conda environments inside of Docker. Though there are people out there who are cringing with me saying that. You can do it inside of a Docker file, and so now you not only have the Python things that you need, you also have the system dependencies that you need as well. So that weird CUDA version for some reason. You can also do things like expose ports. So if you have a web application, the ports can open up, can be opened up, and then you also have the entry point and start command. So these are the commands that run whenever the Docker images are started. Excuse me. Yeah, go ahead.
You said people would cringe if you installed Conda into a Docker environment. What's the alternative, or what would be recommended?
Yeah, if I was giving this talk at DockerCon, I would say you would want to install the packages manually. So like pip is okay. The Conda thing is a little weird because you have to activate the Conda environment. And it's a little bit of a hacky way on how to do that in Docker. Totally works. It's on a lot of my Docker images that are public, but it's the true cold hard Docker people are like, don't use Conda environments, install directly. But the reason for that is there's a chunk of code that you can put at the end of the Docker file that says activate my bolts as an example, activate my bolts2 environment. And whenever someone goes into the Docker image, the bolts2 environment is ready to go, but you can deactivate and get out of it. And the idea is that someone shouldn't be able to like screw themselves over, right? You don't want to get out of the conda environment. So that's mainly the reason why people say that. But sometimes it's without spending up, 10,000 hours on someone's GitHub. They do have a conda environment there, so just use that to get started. Once you have an image built and you run it on your machine, it also can attach to the other parts of your hardware. So in your Docker image, you can also connect to what's called a volume, which is a portion of your file system. So let's say I'm building a Docker image for protein folding, and I have a specific drive on my computer or a specific folder that has all of my sequences. So I can leave all those out of the Docker image and just say, when I run it, connect to my sequences folder and run from there. It also will connect to your memory CPUs and GPUs if you need those. And then also it has the ability to connect to a network, either a network between multiple Docker images or just the network to where you can access the internet. And this is defined in a Docker run command. So whenever you run, you specify what ports you want to open. So in this case, I'm going to map 8888 on my local machine to 8888 inside the container. The same syntax for the volume. I'm going to take my data local folder on my laptop and map it to home user work inside the container. And then I'm going to give it access to my GPUs, and then I'm going to run a Jupyter notebook, and this will open up in a web browser. So let's take a more advanced look. I know, no one hate me. I'm actually going to say bad things here. So AlphaFold 3 has a really terrible set of requirements to run, right? I'm getting some thumbs up. So the scripts themselves are not that big, 20 megs or so. The system dependencies can be upwards of 5 gigs, and that includes the drivers and all the other things that you need to run. That also includes some of the OS system components that you would have locally, but stick with me here. Then you've got about 250 gigs worth of databases, reference databases like Uniref and other things you have to download, make available, and then another gig for just the model weights. So not to shock everyone, but the Docker file for AlphaFold is a little bit longer, a little bit more complex. But one thing I want to point out is up at the top here, they start with the CUDA 12.6 base image from NVIDIA, which is really useful because you don't need to install CUDA in your scripts inside of Docker file, because that's a pain, right? So it's going to start with the version of CUDA that it needs, and then it's going to run on Ubuntu 24.04. I'll skip through some things here, but notice that it will install the Hummer build here, and then also it will run the pip install for about 32 other Python libraries. And then at the end, whenever you run the AlphaFold Docker image, it's supposed to run Python, run alphafold.py. So it's automatically supposed to execute that Python file. So this by itself, when this builds, is 5 gigs, not including the model weights and the databases. So if you wanted to run this in Docker on your local machine or in the cloud or on an HPC environment, you actually need to mount where these other the model weights and the databases are on your local drive. So you're not packaging these up with the whole image. And then the rest of it is just running that alphafold.py script. So a lot of AI models work in this way where you keep your databases and your parameters and things outside of the image. The image just has the stuff that you need to run, and then you mount it, and then you're able to run. Now, why I always say to people, hey, you should start with Docker, avoid writing things in Singularity to start, is because, again, Docker works on your laptop and a bunch of cloud services. But if you wanted to make a Singularity image, for a lot of images, it's just one command. So I can actually take my C438 AlphaFold 3, which is on my Docker Hub website, and I can convert it to a CIF file or the Singularity file format, and then it just runs as well. So this has been heavily used in both its Singularity form as well as the Docker form as well. So this is why it's nice to get things working in an environment on your machine, and then you can convert it if you need to run it on an HPC environment. Now, 15% of the time or so, the conversion is a little wonky, so you have to figure out how to handle that then. But yeah.
Sorry, I don't know what this, but like, how does this file look like? Is it like human readable?
It is, yeah, it is. It's a big file, singular file that you can look inside of it, and it's a little interesting because the CIF file is like a binary representation of what sort of like what the Docker file looks like. So this in of itself is not readable, but the generating the CIF file is just a text file, just like a Docker file is. But again, one thing to remember is that Singularity runs on Linux only. So if you're going to work on your MacBook, you got to kind of do the Docker thing.
So in terms of operations of Docker versus Singularity, the only difference is they are executed by different kind of platforms.
Yeah, and the syntax is a little different. So notice here it's Docker run and GPUs all, whereas in Singularity it's Singularity exec, and this NV is saying load the NVIDIA drivers. So the way it operates is a little bit different, and Singularity operates without root access, so that's why the cluster admins prefer it. So let's say you've got your Docker image up and running, and now you want to run a bunch of jobs, right? So let's say I'm wanting to run my AlphaFold 3 10,000 times. a lot of people overthink what this flow should look like because they're used to the documentation from an HPC website that says, oh, we got to do this weird, like weird logic, right? We don't really need to. How I've done this in the past is just set up a CSV file that has the list of the jobs in it, have a script that loops through each of these records and calls the slurm command of a different script. And all we're doing is we're passing in the parameter here. So let's say fold protein 2PV7 gets passed in here as an argument, and then the rest of the stuff just runs. So all I'm doing is passing in some information rather than passing in, I'm not copying files anywhere, I'm not doing weird things. All I'm doing is just saying, with some parameter arguments, throw that in there, run stuff. Let's take a little simpler example. So bolts 2, which I think more of you like than AlphaFold 3, is a much easier example to run in Docker. So bolts 2, if you look at their GitHub repository, they require that they don't require, but they show in the installer that they use PyTorch 2.7. So you can go grab the PyTorch 2.7 base image, and then also pick the one that has whatever CUDA driver you want to use. In my case, I picked 12.6. You don't really need a whole lot of this. I just kind of made sure that I had access to Git and wget and some other things, and then I just ran the PyPy installer. Thank goodness it's very easy to install, and this just works. So let me go out of this, and I'll show a little quick demo. Maybe not. There we go. So over here in VS Code, I have a Docker. I have the Docker image already pulled on my local machine, but I'll show you what it looks like if I were to try and do this. So if I run this Docker build bolts file, and this is all this is going to do is, well, first I'm in the image here. If I run this, what's going to happen is it's going to go and read this Docker file, which looks exactly like what you saw on the slide, and then it's going to start building. Again, the benefit is because Docker is layered, it doesn't have to go from the beginning, and I built this earlier, so it's actually just going to use the cached versions of my individual things here. So what happens is if I were to go into the Docker file and say, oh, I actually don't need curl, just take that out, no big deal. Now what's going to happen is, what should happen is my first layer, which is the PyTorch base image, it should keep that cached. Now it's going to have to rerun the second layer because I changed something. And then now the third layer should also be changed because the order matters in these. So if I go back and try and rerun this, now you can see that it's going to run that apt-get again, leave out curl this time, and then it will then run that the if install bolts section as well. I won't bore you with this, having that do that. But one thing to keep in mind is that there is a huge set of publicly available images on what's called Docker Hub. So Docker Hub is really useful because it will allow you to pull a lot of base images from there. PyTorch is very heavily maintained on there. You can get all sorts of different versions, things like TensorFlow, and also other servers like Nginx, Jupyter Notebooks, etc. All this is available on Docker Hub. But I also host a lot of the images on here too. So like you can see my bolts image for 2.1.1, and then I also have a version with weights in it. Don't do that, but I did it for this demo. And you can grab those from here. So in your case, if you wanted to use my bolts image, you just do Docker, click the right one here. Show it here. All you do is just say Docker pull, and it will then pull, it just downloads it without having to actually rebuild it on your local machine. So again, don't reinvent the wheel. See if what you're trying to run has already been done by someone else. If it's a really cool AI biomolecular model, I probably have already built the Docker image for it, unless it's really terrible. And so check there and see how it works. Yeah.
Let's say I build one model a day, and I want to build another one next week, and I keep doing this for every before, well, what kind of machine would I do, run it on? Would I try to find some place in the cloud to do that, or would I buy another laptop?
Yeah, so luckily Docker, if you push your Docker containers to Docker Hub, I don't know what the limit is for free storage, but I haven't hit it yet, and I have a bunch of images up there. But luckily on the Docker file itself is just a text file, so it's not hosting very much. But on Docker Desktop, which is the GUI, I can actually go through here and delete my old containers that I'm not running. So for example, let's say you made something a month ago, you used it then, it's taking up 10 gigs of space on your laptop, and now you're done with it, you can delete it from here. And you can either leave it stored on your Docker Hub for free, or if you just needed it again, you would just rebuild it from the Docker file, which is just a text file. So it's kind of it's your prerogative as to where you want to actually run it. So to run these locally, I'll try not to bore you too much with how this works locally, but if I wanted to run this locally, I just use the Docker run command. In this situation, I'm mounting the folder that I'm in just because I have this protein YAML file. I'm giving the container a name, so it says I'm calling it bolts. This rm command says remove the container if I go out of it. This is really useful if you don't want to have to remember to delete the container when you're done. IT means interactive, so give me an interactive shell to run in. So you can see here that I'm actually in the container. So notice that I was in my Colby at Tuple MacBook machine first. I went into the container, and now I'm at this root at whatever. And then I can go and change directory to the mount location and ls, and now you see everything that's in that folder. So because this is actually connected to my local volume, I can create a file inside the container and it shows up outside, which I think is really useful, especially if you're running these models. And then if I want to, in this case, want to run bolts, I can just do bolts predict protein.yaml, and this should kick off and start running in just a second. I have no GPU on this MacBook, so bear with it. Yeah, so anyway, it's starting to run. Because I ran it earlier, it ceased, but you can see the results that it produced. Maybe. Long suspenseful pause. All right. So just 4 pro tips for working with containers to kind of stop this section.
Just one quick
Yeah.
question. How does it work if you don't have a GPU there?
Yeah, if you don't have a GPU, for like the PyTorch models, most of the time in the scripts, there's a command that says look for the GPU and either say that the device is CPU if it can't find one, or else default to the GPU. So the AI models usually will run locally, they're just way slower. So like if I run that bolts model, even on a small protein, it takes like 10 minutes, whereas it would have taken 30 seconds or something on a GPU. So a lot of times this is where if you get your code working locally, when you push it up to a different environment that has more horsepower, a GPU, more RAM, whatever, it's usually the same. It's just slower on your local machine. Any other questions before I go into my tips? Okay. So pick the closest base image. Don't try and reinstall a CUDA driver yourself in your Docker file. Be kind to yourself. And also you can pick specific versions of things like PyTorch or CUDA itself. Leave out the data sets and model weights because that will keep your Docker image as small as possible. So you can still have the data or the weights and things on your local system and you just mount to it and run it from there, no big deal. And along that same vein, pack light. And what I mean by this is if in your Docker file, you need things like sudo or wget or whatever, that's cool, put those in there. But there's no need to put a bunch of other stuff in there as well. Don't bloat the image with other things. In fact, sometimes that causes groups that want to run your model to flag it because it has too much stuff in it, like security vulnerabilities and all that kind of stuff. So just leave the stuff out that you don't think you're gonna need. And then the fourth is build one container for one task. So in this situation, the bolts container just runs bolts. It does not have the MSA server inside it. That should be run either through Sergey's beautiful API or as a separate Docker container that's running the API itself. So one container, one task. The reason for this is it's difficult if a task were to fail. So like if I were to spin up a database and spin up like some like Jupyter Notebook or something, and one of them were to crash, it's very difficult to restart a process inside the container after it's crashed. The idea is like if something screws up, just blow away the Docker container and rerun, right? Turn it off and turn it back on again is what we're after. And it's very difficult to do that if you bundle too many things inside the Docker image. So in this case, don't bundle your tool with like an IDE. So don't put bolts inside with Jupyter Notebook in there. Avoid that. All right, so now that we've got our beautiful Docker container up and running, where do we use it? And where do we use it once we've hit the limits of our local laptop, right? So for the last part of this, I'll talk about the cloud, and I'll talk about HPC environments and how do we scale these molecular experiments. So obviously I'm a little biased. I'm a cloud engineer by training. So I use the cloud a lot, but I'll give you some benefits that I think that are really useful for the science world. We get elastic scalability, meaning I can configure my cloud services to grow with whatever my needs are. Meaning I can set up a cluster, I can hit the cluster with 1,000 experiments, it balloons out to handle that workload, and then spins itself back down to almost nothing. So this really helps to where I'm not spinning up 1,000 servers and leaving them up and running and I have to remember to turn them off. A lot of the newer cloud services are just elastically scalable. And then we can orchestrate complex data tasks. This is a perfect thing for what we're talking about with the biomolecular modeling. And then lastly, I think this, by putting things in a cloud environment, is a lot easier to collaborate and share things with your collaborators, and then also because of the way things are run in the cloud, I think it's easier for scientific reproducibility. It's easier for me to say, this is my Docker image that I ran my thing in, that I trained my model with. So if somebody wants to reproduce that, they just run your Docker image, right? Rather than having to go back and make sure the Conda environment's the same as what it said in the install file, and oh wait, that CUDA driver doesn't exist anymore because NVIDIA's cheap, even though they just hit 4 trillion in valuation, or little things like that that are always gotchas whenever we're talking about scientific reproducibility. At the core of a lot of cloud architectures, we use OneDrive. Oh, sorry, not OneDrive, excuse me. We use OneLake or data lakes. Data lakes are sort of amorphous file storage where we can put lots of things. So this is an example slide from bioinformatics workflows where we store data in its various forms. So you can see here, I've organized the data lake to include different types of data, exome data, pathology images, phenotype information. And you could start doing the same thing, thinking about how you want to organize your experimental data. And you may think, well, as long as I know where it is, why do I care if it's organized? You sound like a hoarder. But secondly, it also makes it really easy whenever you organize things well for the other compute services in the cloud to operate across that data. So as an example, if I have my data organized in a way that it makes sense, where I've got an experiment, I've got an experiment set, I've got a target, I've got a particular assay, I've got whatever, and I've organized things accordingly, then I can ask questions like, give me all the small molecule candidates in experiment set B that have a binding affinity of less than 2 nanomolar. And I can ask these sort of queries across my data a lot easier rather than having to go and like grab all that stuff, pull it down locally, hope and pray that that Excel file opens and that no one screwed it up, and start organizing that data that way. There's also other examples here of some other queries. So I've used the word Kubernetes about half a dozen times, but what is it? So Kubernetes is an open source platform developed by Google that it uses Docker containers and it will spin up compute nodes as needed. So it's sort of like, how do you run multiple Docker containers in a cluster environment? How it works is that you specify different resource needs by a pool, and then those resources could be things like how much memory do you want, how many CPU cores, how many GPUs, etc. So don't worry, there's not a quiz after this. But the way that this looks inside of a Kubernetes cluster is that we specify the node pools. So for example, this node pool could be my high memory node pool. And within that, when I submit a Docker container to be run in there, it spins up in a node that has the appropriate amount of horsepower left to be shared. So a Docker image is run in what's called a pod. And multiple pods can be run inside a node if they can share it. So for example, if my node has 512 gigs of RAM and I have a job that just needs 100 gigs, I can run multiple things in that same environment, assuming you have enough resources. But a lot of times for these bigger workloads, we sort of have a one-to-one relationship where the one Docker image is running in one pod, which is taking up the whole node and using everything that that node has to offer. Yeah.
How do you sort of estimate like how much memory you need to allocate per node?
Yeah. The big thing is knowing what you're running. So for example, if you're running something like a large language model, for example, and you know how many parameters it has, you sort of take that number and multiply it by 2, and you need a GPU that has that much RAM, as an example, right? So like the Microsoft B model that I was talking about earlier has 14 billion parameters. It takes about 30 gigs of virtual RAM on the GPU to inference through it, to tune through it and inference. So there's some like napkin math calculations that you can do, but as a principle, if you're running things in like Python or R, those are memory heavy languages. And so if you're loading in a 1 terabyte data set, you've got to have a terabyte of RAM to handle that, as an example, right? And then a lot of times with CPU cores, it's if your code is able to be parallelized across multiple cores, then the more cores, the merrier. But if you have something that's just a Python script that is not parallelized, you don't need to give it 48 cores because it's just using the 1, right? So you sort of have to like Goldilocks size it a little bit based off of what you're running. So how do we define these things? So inside, so when I want to make a pod spin up with a Docker image, this is what the pod spec looks like. So I give it a name for the individual pod. I also tell it what the Docker image is, and you'll see a actual example in a minute. I also tell it what data volumes I want to mount. So if you have your data in a data lake, I can mount that data lake just as part of the file system, and it operates normally just like it's part of the file system. And I don't have to worry about the underlying service architecture or anything like that. I also can specify the command and arguments as well. So the command would be something like, what shell do I want, or do I want to run Python to start? And then what arguments do I actually want to give it? Like, do I want to run the Python file, or do I want to run some sort of loop or whatever? You also can define the resource limits. So this is where if you need something if you need to run something that has a lot of that requires a lot of RAM and a lot of CPU cores, you can define that here as well. And then there's also a couple other things. You can also give it access to secrets. So let's say you inside the container, it's going to hit some sort of API, and the API has a secret. I don't want to pass that around in free text. I can pass that in here. There's also the ability of what's called node affinity, which says that, all right, I've got this type of workload, and I got different pools that could possibly support it. And Kubernetes is pretty smart as to assigning what type of job can go in what type of pool. And so there's the attraction affinity is called tolerations, which sounds like a Southerner said, made that like what in tarnation. So the tolerations actually give you an attraction to a particular type of node. And then the opposite is called a taint. Why they called it that, I don't know. But that's the repelling version to say, I can define a taint that says, don't put this on a GPU box, even because I'm not going to use a GPU. And then the attraction thing says, you run it on an AMD EPYC box because I know that this runs really well. And it needs to have 32 cores. And the big benefit is that Kubernetes auto-scales. So this is a screenshot of an RNA-seq process that I've done where I submit hundreds of jobs, and then all of a sudden the pods start popping up. So let me show you what that looks like in a live demo. Pause for suspense. There we go. And so over in my Azure environment, I have a file system that I have, or a file share that I created. I have a Haddock 3 experiment here. So Haddock, if you've never used that, that's this molecular docking tool that I use in a lot of my research. It has a config file here, and then it has some data inside here that has the PDB files that need to be docked. So I have that set up in my file share. And then over in my Kubernetes environment, you can see that I have a pod spun up. if I can read this, you can see that I have a Haddock 3 pod spun up here and it is running. How I got that to spin up is that I have a Haddock 3 pod file or a YAML file here that says, call it Haddock 3 pod 001, pull my Haddock 3 image and mount my data to it. And this little command here is the hacky way of saying, just keep it up and running, because I'm running a demo, please don't turn off while I'm running a demo. And so, but I could also have passed in the Haddock 3 command here, or something like bolts, or any of the system commands can go here as well. So while I'm in my terminal, I can remote into that container, that pod that's running, because I number there. Beautiful. So now I'm actually inside that that pod that's running, and I can actually go in and look at that mounted data set. And you can see here I've got the Haddock 3 folder that I just showed you on the web. So inside, there we go. Okay, cool. So last demo that I'll show is, in addition to so you may be wondering, how do I pay for cloud stuff, right? We're all kind of scared, especially in the academic world where you have limited money. How do you run things in the cloud environment, and how do you pay for them? So for things like Kubernetes or virtual machines or any of these things, you pay for it while it's up. So for example, if I'm running a GPU box that's 20 bucks an hour and I'm running it for 4 hours, that's $80 and that will get charged to you. But if you accidentally leave something running, then you also get charged for it. Microsoft doesn't care if you're an idiot or not, like they're gonna get their money. And so the way that the benefit of the cloud is that there are now services that are serverless. So one of those is called container apps. I'll show you a very quick demo of this in just a second, but it's a serverless infrastructure. It's Kubernetes under the hood, but without the need to actually manage that infrastructure. So I'm not managing any of the networking, I'm not managing any of the boxes, I'm just submitting jobs to it, they run, and then I get charged for while they're running. So the demo that I'll show on this is around GUI-backed apps, because I think this is a really good use case. So let's say you are the person who developed Bolts 2 or Protein NPN, which is the demo that I have here. There's on Hugging Face, there's a Hugging Face space for Protein NPN. It's actually, when you create a space on Hugging Face, it actually builds a Docker image. And then over here in my container, the container app environment, this is the thing that's going to run the serverless GPU. I actually can just point to that Docker image, or any Docker image that it has access to. And then what happens is now Protein NPN is running on in that container app environment. So I can run my demo here. It's the compute's turned off because I've waited too long, and then it will output the response here. I can also, if you're fancy and you have a lab URL, right, I can also connect this to my own URL. So there's my domain name with this Protein NPN hosted at it. And the benefit of this is that when something's not running, the container itself will spin down to 0. So you can actually see here, because I've been a little long-winded, that because I wasn't running anything on this container, it's now scaled down to 0 nodes, and I'm not getting charged for it. And then whenever someone starts using the thing, let's say all of you went to the website at once, this would then scale up accordingly so that it meets the needs of the traffic. So with serverless, to kind of close out here, with the serverless technologies, there's a couple different services across the cloud vendors that do this, that allow you to run things like APIs, apps, etc., with a GPU backend or with a CPU backend, whatever. And Azure automatically handles the infrastructure and scales it accordingly based off the limits that you set. And it simplifies the operation. So you don't have to be a cloud architect that's managing networking, right? You don't have to be a security architect that's keeping track of that. So it's perfect for the scientific people who just need to run things, right? And it allows you to get started a lot quicker with that managed infrastructure. And then lastly, you get some cost savings because especially for applications that aren't being used by 10,000 people, right? It's a research app that might be used by one person in a week. But when that one person needs it for their dissertation that's due tomorrow, they really need it, right? And it's available enough. So to fold it all together, I hope that I've, in my long-winded talk here, I've given a little bit of insight into how we can start building models even though we don't have a whole lot of data. Tuning works pretty well. There's lots of things we can do to the limited data that we have, and you can see that we're starting to see some pretty good results from that Polike project that I mentioned, even on 5,000 records of data. Once I have this stuff up and running, or once I'm scrolling my LinkedIn and I see some new model that I want to play with, I can build Docker images that allow for me to containerize and make that code modular, make it run on my laptop, but also make it run in someone else's environment or on the cloud without screwing up my local environment. And then lastly, I can deploy these things to an HPC environment if I want to run multiple experiments, or I can also send them to a cloud cluster environment like Kubernetes, or even host the UIs in some other serverless architecture as well. And a benefit of the cloud is that it does give you that flexible scale for kind of external facing things that you need to share with other people. So model, package, deploy, repeat. And with that, I'll take the other questions.
Can you talk a little bit about like container orchestration, like with multiple containers?
Yeah.
How to do that?
Yeah, so in the case here, there's in that YAML file where I submitted the Haddock job, I could also, let's say that was a Boltz demo instead. So with Boltz, as an example here, I have a pod for Boltz as well. In the Boltz command, I could actually say to run the predict command, and in this situation, I'm saying use Sergey's MSA server, right? But let's say you wanted to host your own. You could have a second Docker container that's running on a separate pod that this can communicate with. Because the Kubernetes cluster itself has a network, each pod has its own IP address, and so I could have just said use MSA server and then given it the IP address of the other container that's running. So you can orchestrate different running things inside the same environment. Yeah, you and then you, or either one.
Yeah, I'm used to Slurm.
Yeah.
I've never used Azure Kubernetes. Is it just different frameworks, same sort of end goal?
Yeah, the difference is with Slurm is you're scheduling against other people and your own quotas, right? So Slurm is really great because you're saying, here's my jobs, here are the requirements that I have, right? I want a GPU box, I need 15 cores, whatever, and it works. The difference is, and we've all experienced this, where you don't have access to the boxes that you want and you're waiting in a queue for forever. The difference in a cloud environment is that you just submit it and it just spins up and runs and you're just billed accordingly for it, right? So it's not really a schedule, there's no scheduler because there's nothing to schedule because things are just ready to go.
AWS that I've worked with runs Slurm.
Yeah, there are certain HPC cloud services for groups that want to continue running Slurm. Have the
Like Spot
yeah
GPUs and that sort of.
yeah Spot GPUs, and then there's one called Azure HPC services or Cycle Cloud. So there are HPC services in AWS and Azure, whatever, that mimic an on-prem HPC environment. Okay. They do that for academics that don't want to change to Kubernetes, but not really. But they do that because you can still control billing and stuff in a way that IT is happy with, right? So you can totally just do a Slurm cluster on Azure or on AWS, but you can also do like the Kubernetes environment where in that situation, like your PI can set up a budget for the whole subscription and you're just kind of hammering toward that the budget. It's a little bit different paradigm. Yeah, good.
How do I know if like whatever language model I want to tune is tunable on Hugging Face?
Yeah, so on Hugging Face, it actually tells you what the framework is. So here, well, any model is tunable if you can do it manually, right? But if you wanted to use the transformers package that I mentioned, there's a flag here. So let's say I wanted text generation models and I want it to be compatible with the transformers framework and I want to use PyTorch. Now you're starting to see it filtered down to that set. So any of these are able to be tuned with just a few lines of code from transformers. And it's a lot, 68,000. So The diffusion stuff is a little different. So protein diffusion models are not supported by any of these sorts of things because the way we do like SE3 equivariance and stuff is not like generating pixels. So none of the protein diffusion models are supported in any of these frameworks yet. If you want to do image diffusion, you can also tune those as well with the same sort of, it's called diffusers is the equivalent to that.
So what were the results for your tuning of V4? They good enough for you?
Not yet.
Okay.
Not yet. They were good enough to make a slide for a talk, but they're not good enough yet. What's happened is, and I hinted at this, is that the training data, the sequences for the heavy and light chains are sometimes the full fab, sometimes the FV portion, and then sometimes something in between or a little bit more. So what you actually saw with that figure, what you didn't see with that figure is I chopped to just the FV portion, which looked pretty good. If you zoom in, some of the secondary structure of like some of the CDR loops and stuff is not quite right yet. But it still was able, all of them were still able to be numbered. It's just, we're an N of 4 here, but they still work good. But then the stuff that's beyond the FV was a little swirly, which I chopped out for TV purposes. So we're still working on it. Yeah.
In your work, when you were using diffusion model, so what was your approach in order to develop those new molecules or the antigen or antibodies?
Yeah, so for the PD1 and H5, HA1 proteins, the diffusion model was EvoDiff from Microsoft, but I use it in a slightly different way than how their README shows. I actually diffuse with the proteins in place, so the antigen and the antibody, and then just select the CDR loops and diffuse with it in place because it will take structure information. And it's actually called in-painting is their mode. So it's a little different than how they do it. For the EGFR challenge, I used a blend of EvoDiff, but also ESM has some diffusion capabilities as well, or infilling capabilities as well. It didn't work as well as I would want it to, but those are the two models there.
In your work, have you ever developed your own diffusion model from scratch?
I have not, and that's something for since we're going to have this data set of antibody and antigen sequences that we're curating, I'm hoping to do something like a diffusion model out of that as well. So different than a PLM.
One more question. Yeah, sorry for that. As I said, if you want to develop a molecule where we target that specific idea region, so where we can find that kind of data, because I literally searched 1.5 months and I didn't find enough data where I can train my diffusion model. Yeah,
if you find that, let me know. That's the problem. What we're doing for the Polike project, the PLM, is that I have a script that I wrote forever ago that finds polar contacts between chains. We're using that as contact information. There's another library that came out. It's called like Panda something, that not pandas, but it's another package. It's Panda something, Panda Probe or something, that includes other types of biomolecular interactions, so pi bonds and other electrostatic interactions and stuff. And that's what my student is currently trying to see is that going to work for highlighting epitope or active residues in the sequence. So what I'm hoping is that, so this project is halfway to make a new model, but it's also halfway to just like curate a nice data set that other people can use. And so we're still testing out the different tools to do that.
Thank you.
All right, let's thank Colby again. So Colby will be here. You want to pepper him with your questions? Sure. Let's see. So Sergey, next meeting we have a talk. We'll be back at MIT next month. We have a speech friendly line.
It's Martin, Minecraft guy.
Oh, okay, Martin from Bruno's group.
Minecraft?
Sorry,
Yeah,
Minecraft.
So Martin will be talking about Minecraft next.
We might also talk about Minecraft. I don't know.